Hello! In this video you will learn about the workflow of DNA sample preparation for next-generation sequencing with MGI platform. Before DNA or RNA samples can be sequenced by next-generation sequencing, they must be fragmented and repaired and collected into adapter-ligated libraries. Preparation of samples can greatly influence the sequencing results. Short-read sequencing technologies like MGI cannot sequence long fragments. Therefore, the first step of sample preparation is fragmentation. The protocol is suitable for preparing various lengths of libraries. The necessary length of the library depends on sequencing strategy. The longer the sequencing length, the longer the fragment should be. DNA fragmentation can be done either using enzymatic or physical methods, adjusting the fragmentation conditions based on the target size. We use physical fragmentation with ultrasound. Sonication subjects DNA to acoustic cavitation and hydrodynamic shearing by exposure to brief periods of sonication. Based on necessary sequencing length, the optimum fragment length can slightly vary, but mostly it's 350 to 450 base pairs. The right conditions should be selected for the target length. For 150 base pair sequencing, we use from 400 base pairs long fragments, so the conditions for sonication are chosen from the table below. "300 nanograms and 55 microliters of sample are pipetted in Covaris microtubes, which are compatible with the S220 system and are meant for small sample volumes and fragmentation size. After the fragmentation is done, there is still a high quantity of fragments that do not fall in the target range and will interfere with the sequencing chemistry. Therefore, it is necessary to get rid of these fragments by performing size selection. Size selection is required to produce a uniform distribution of fragments around an average size. It is done by using magnetic beads. Alternatively, also separation with gel can be used. Magnetic beads are paramagnetic, meaning they are magnetic only in a magnetic field, and this prevents them from clumping and falling out of solution. Each bead is made of polystyrene surrounded by a layer of magnetite, which is coated with carboxyl molecules. The carboxyl group is activated in the presence of acid solution. A hydrogen on the carboxyl group becomes positively charged, thus attracting the negatively charged DNA. Larger DNA molecules have larger charge. Therefore, by changing the bead to DNA ratio, we can select different target sizes. 50 microliters of DNA sample plus 50 microliters of beads will give a bead to DNA ratio of 1. General rule is, the lower the beads to DNA ratio, the larger the final fragments will be at elution. You can study the table on the screen. Smaller fragments are retained in the buffer and usually discarded. Please keep in mind that magnetic beads must be stored at room temperature for at least 30 minutes and carefully suspended before use. We use all 55 microliters of the fragmented DNA sample and dilute it up to 100 microliters in a low bind 1.5 milliliter tube. In order to obtain the target fragment size of 420 base pairs, we add 60 microliters of magnetic beads to the 100 microliters of the DNA sample and mix by pipetting at least 10 times. After 5 minutes of incubation at room temperature, the tube is placed on a magnetic rack for 2 to 5 minutes. Then the supernatant is carefully transferred to a new tube without disturbing the beads. Tubes containing the leftover beads together with fragments that are too long are discarded. In order to perform the double size selection, the supernatant from the previous size selection is mixed with the second corresponding volume of beads, which is 20 microliters at this time. After 5 minutes of incubation at room temperature and 2 to 5 minutes on the magnetic rack, the supernatant, together with fragments that are too short, are carefully discarded without disturbing the beads. After the double size selection, the microcentrifuge tube must be kept on a magnetic separation rack. The beads are washed with 200 microliters of freshly prepared 80% ethanol. After about 30 seconds, ethanol must be discarded, and the washing step needs to be repeated one more time. When the second washing is done, try to remove all the liquid and air dry the bead with the microcentrifuge lid open. Beads are dry when they no longer appear shiny, but before the bead pellet starts to crack. At this point, you can take off the low-bind 1.5 ml tube from the magnetic rack and dilute the DNA in 32 microliters of TE buffer. TE buffer is made of tris, a common pH buffer, and EDTA. It is provided in the kit. Take into account that during the double size selection, 60 to 95% of initial DNA in the sample is usually lost. After this step, it's important to normalize the amount of DNA so that all samples would have the same input for the next step. Concentration of each sample is measured using qubit fluorometer and high-sensitivity double-stranded DNA assay kit. Please look at the figure. The fluorometer is used to determine the concentration of the target molecule in the analyzed sample based on the fluorescence emission. The fluorescent dye emits light only after binding to the target molecule. The fluorometer is calibrated using the standards included in the qubit reagent kit, of which standard 1 does not contain DNA, but standard 2 contains 20 nanograms per milliliter DNA, defining the measurement range. By reading the fluorescence intensity in standards with a predefined nucleic acid concentration, the qubit fluorometer creates a straight line against which the concentrations of the target molecule in the analyzed samples are determined. 50 nanograms in volume that does not exceed 40 microliters is the recommended amount of DNA. However, the protocol is suitable for as small amounts as 5 nanograms. If smaller amounts are used, several adjustments to protocol should be made according to protocol. When doing calculations, please use the following formula. 50 nanograms of the fragmented size-selected sample from the previous step is transferred to a new 200 microliter PCR tube. After previous steps, the ends of the fragments are uneven. Therefore, end repair is performed. During end repair, DNA polymerase is making the fragments' ends blunt and even. After the blunt ends are made, the poly A tail is added, which will serve as attachment site for adapters, since adapters contain thymine overhang on one end. To make an ERAT mix, you need to combine 7.1 microliters of ERAT buffer with 2.9 microliters of ERAT enzyme mix for each sample on ice. For example, if you want to work with 10 samples, you can multiply each of the components with 10.5. 0.5 will be in reserve for pipetting errors. So you combine 74.6 microliters of ERAT buffer with 30.5 microliters of ERAT enzyme mix. Mix it thoroughly, and then 10 microliters of ERAT mix are added to each of the samples. The volume on how to prepare the ERAT mix is shown in Table 3. From this step forward, when adding reagent to the samples, enzymes and samples need to be placed on ice, except for bead cleanups. After adding the ERAT mix, samples are vortexed 3 times for 3 seconds each time, centrifuged and incubated according to the protocol. Samples are placed in the thermal cycler in the following conditions. Before choosing the adapters, please read the Appendix D of the protocol carefully. Adapters are added based on sequencing strategy. Samples that will be sequenced on the same lane must have a unique adapter each. In addition, a full set of adapters needs to be used among all samples within one lane. The sequencing flow cell consists of 4 lanes. Adapters may overlap between different lanes, though not between different samples within one lane. Five microliters of the adapter have to be added to the 50 microliters of reaction mix from the previous step. If a smaller amount than 50 nanograms of sample was used in the beginning of the protocol, then adapters need to be diluted accordingly. For example, if 25 nanograms of the sample were used, then the adapter must be diluted 2 times. During this step, a ligase enzyme covalently links the adapter and DNA fragment. Adapter contains a reference code, which allows to identify each fragment from the same sample. Adapters allow multiple samples to be sequenced together on the same flow cell. Adapter also indicates where the sequencing primer should bind, and sequencing should begin. To make a ligation mix, it is needed to combine 23.4 microliters of ligation buffer with 1.6 microliters of DNA ligase for each sample. While preparing a ligation mix, it is important to remember to pipette very slowly due to the viscosity of the ligation buffer, and to place reagents and samples on ice. When 5 microliters of adapters and 25 microliters of ligation mix is added to the reaction mix, samples are vortexed 6 times for 3 seconds each time and centrifuged. Samples are placed in a thermal cycler in the following conditions. There are two options depending on which magnetic separation rack you have. If you have a 96 well magnetic rack, it is possible to leave samples in a 0.2 ml PCR tube and perform a cleanup. But if only a 16 well separation rack is available, then you need to transfer samples to new 1.5 ml low bind tubes. After the reaction is finished, 20 microliters of TE buffer is added to each sample in order to reach the total volume of 100 microliters. After that, ligation products are cleaned up by adding 50 microliters of magnetic beads and subsequently washed twice with 80% ethanol, as previously described. Polymerase chain reaction is performed in order to amplify the target fragment. Extremely high fidelity is essential to limit PCR errors that may result in false variant calls in sequencing. During the PCR step, the target fragment is multiplied many times by 3 processes being repeated: denaturation, annealing, and extension. During denaturation step, double helix is unwinded in 2 separate single-stranded DNA strands. During annealing phase, primers are annealing to the single-stranded DNA. During extension or elongation phase, in this step, the polymerase enzyme sequentially adds bases to the 3' end of each primer, extending the DNA sequence in the 5' to 3' direction. After each cycle, the amount of DNA is doubled, and the overall amount is growing exponentially. For the PCR step, it is necessary to take 19 microliters of the sample from the previous step. The rest of the 19 microliters are saved as a backup if elution was done in 40 microliters of TE buffer. PCR reaction mix is made from 25 microliters of PCR enzyme mix and 6 microliters of PCR primer mix for each sample, plus the reserve for pipetting errors. For these 19 microliters of each sample, 31 microliter of the PCR reaction mix is added. Samples are vortexed 3 times for 3 seconds each time, then centrifuged, and PCR reaction is set in the following conditions. Please note, If the amount of sample used in the beginning of this protocol was smaller than 50 nanograms, then amplification cycles are adjusted according to the Appendix E of the protocol. After the PCR step, another cleanup is performed according to the previous description. After the bead-based purification of the PCR product, the concentration is measured with cubit fluorometer and high-sensitivity double-stranded DNA assay kit. The required yield for PCR products is more or equal to 1 picomoles per lane. The concentration of the target molecule in the analyzed sample is determined by means of a fluorometer compared to a known concentration in the standards. The method is based on fluorescence emission. The fluorescent dye emits light only after binding to the target molecule, which in this case is DNA. In addition, the overall quality of the fragments is visualized with Agilent Bioanalyzer. The peak should be observed at the target length, should be around 504 base pairs. The method is based on capillary electrophoresis on the chip. Fluorescent dyes are used which emit a fluorescent signal when binding to DNA or RNA. Under the influence of the electric field, negatively charged DNA and RNA molecules move through the polymer filled in the chip in the direction of the positive charge at a different speed, which is determined by the length of the fragment. Length markers are used to determine the exact length of the fragments, which in the figure is peak 1 at the beginning and the second one at the end. After the quantification, another normalization can be done in order to ensure that for the rest of the steps the same amount of DNA will be used across all of the samples. Depending on the concentration of each sample, the samples that are intended to be sequenced on one lane are pooled together so that the total amount of DNA on one lane reaches 1 picomole and does not exceed 48 microliters. Let's say we are going to sequence only one sample per one lane. The concentration of the DNA sample after PCR is 40 nanograms per microliter. Using Bioanalyzer, the average length of the fragments of the samples is detected as 504 base pairs. To calculate how much of the sample corresponds to 1 picomole, we need to use the following formula, where the fragment size is divided by 1000 and multiplied with 660. The conclusion is that we would need 330 nanograms of the sample in order to reach 1 picomole. To know how many microliters corresponds to 330 nanograms of our sample, we need to divide 330 by the post-PCR concentration, which was 40 nanograms per microliter. So in this case, we must take 7.4 microliters of the sample for the circularization. If we need to sequence more than one sample per lane, we must pool the samples together. To obtain the necessary volume for pooling, we need to do the same calculations for each sample separately, and afterwards divide the final volume with the number of samples that are planned to be sequenced on one lane. This calculation will be suitable for cases where you expect an equal amount of data to be generated for each sample. If the total volume of the pooled samples is less than 48 microliters, then TE buffer is added to reach this volume. But note, if it exceeds 48 microliters, the pool should be concentrated to reach the right volume. Circularization is started with the denaturation stage, during which DNA strands are unwinded. We transfer 48 microliters of pooled samples in a 0.2 microliter PCR tube and put it in the thermal cycler in the following conditions. It is important to place the samples on ice immediately after the incubation in order not to let the strands bind together again. Prepare the following mix for a circularization reaction by combining 11.6 microliter of splint buffer with 0.5 microliters of DNA rapid ligase for each sample. Splint buffer from this step contains oligos, which binds to the sequence adapter and serves also as a starting point for DNA rapid ligase to attach and start copying the fragment. After this reaction, DNA fragments will be circular, so while DNA strands are unwinded from the previous step, 12.1 microliters of circularization mix is added, and samples are vortexed 3 times for 3 seconds each. Then the reaction mix is placed in the thermal cycler in the following conditions. Circularization is followed by enzymatic digestion, during which the fragments that were not circularized are digested. It is possible because the digestion enzyme has exonuclease activity that is digesting linear DNA. To make an enzymatic digestion mix, 1.4 microliters of digestion buffer with 2.4 microliters of digestion enzyme for each sample needs to be combined. Then 4 microliters of enzymatic digestion mix is added to the circular products from the previous step. Samples are vortexed for 3 times 3 seconds each and centrifuged. After that, samples are placed in the thermal cycler in the following conditions. It is important that immediately after the reaction is finished, 7.5 microliters of digestion stop buffer is added to the samples to stop the digestion reaction. After the stop reaction, samples need to be transferred to new 1.5 ml low bind tubes, and another cleanup with magnetic beads is performed as previously described. The only differences this time are extended incubation time for 10 minutes and increased washing volume, 500 microliters of 80% ethanol. The quality of these samples is determined by quantifying the purified enzymatic digestion product with qubit and single-stranded DNA assay kit. The ratio between the final enzymatic digestion products and input products of PCR should be more than or equal to 7%. So if the sample input was 50 nanograms, then the single-stranded DNA concentration should be at least 3.5 nanograms in 1 microliter. Thank you for watching! The creation of this video was sponsored by Integramed project.